Right, this video is going to be about our mitosis models. So for our mitosis models, I'm going to start over here with this model over here. And so this model, before we get started, I'm going to kind of go over, this is the cell membrane. This whole area here is the cytoplasm, which kind of just includes the cytosol and the organelles and any inclusions that are within the cell. So on these models, you can't see the organelles specifically, they're just not drawn. Mitosis is going to be happening, is what happens in the nucleus primarily, so that's what we're going to be focusing on. In here, we have these are going to be our chromosomes. So our chromosomes have condensed within the nucleus. So here's our nucleus, and you can see that that these chromosomes are starting to condense. This little guy right here in red is the nucleolus. And then these guys here, these dark black guys, are going to be the centrosomes. And so these have already duplicated, and they're going to be starting to move to opposite poles of the cell, as you can see here. Around here, they've got little star asters that are radiating out around them. So this is going to be very early prophase. So we can see that it's prophase because our chromosomes have started to condense. And so before it would have been very sort of fluffy chromatin, but they've kind of started to condense here in chromosomes. We now have our duplicated centrosome. So we have two of them. So remember, a centrosome is made up of two paired centrioles. So each of those would have two centrioles. So we kind of have four total. So we have two centrosomes, and each centrosome is made up of two centrioles. And so they have just been duplicated, and that was finished in the G2 phase of interphase. So this is probably moving right from the G2 phase of interphase, and then prophase is just starting once these chromosomes start condensing. So that's going to be very early prophase. Next, we're going to move here to sort of a middle prophase. And so this, we can see that the chromosomes have condensed even further. You can see individual little chromatids in here. The centrosomes are starting to move to opposite poles of the cell. And then these here, this is the mitotic spindle, and it's made of spindle fibers, sort of microtubules that are going to be important in directing mitosis. We still have this area here, this sort of light gray color is the nuclear envelope, and we saw the nuclear envelope was still here. So the nuclear envelope is intact in both of these two. As we move a little bit further into prophase, so this is going to be our late prophase. And some texts call this prometaphase. The nuclear envelope has actually dissolved at this point. And so the nuclear envelope has now dissolved. We have our centrosomes at opposite poles of the cell. And then you can see these little red guys right here. These are the spindle fibers that are now going to start attaching to the centromeres of our sister chromatids. As we continue, we move from prophase or prometaphase into metaphase. And so in metaphase, all of our sister chromatids are lining up along the middle of the cell at what's called the metaphase plate or the equatorial plate. We still have our centrosomes on opposite poles. You can see this whole pink area here is the mitotic spindle, and you can see that these individual spindle fibers are going to be attached to the centromeres that join those sister chromosomes, the sister chromatids. And so this here is going to be metaphase. This is the metaphase plate. Here are our sister chromatids, and the sister chromatids are going to be attached to these mitotic spindles. All right, after metaphase, we move into anaphase. So this one here is going to be early anaphase. So if you noticed before in metaphase, all of these sister chromatids were attached together at the centromeres, but now the centromeres have been pulled apart, and the sister chromatids are being pulled apart, And these spindle fibers are pulling the sister chromatids to opposite sides of the cell. And so we can see the whole part over here is the mitotic spindle. These are the spindle fibers that are pulling the sister chromatids to opposite sides of the cell. That's early anaphase. This is a little bit later anaphase. We can see it's these sister chromatids have made a little bit more progress and are moving further and further towards their sides of the cell. The other thing we can see here is this right here, E, is called the cleavage furrow. This is going to be part of what we call cytokinesis. So mitosis was everything happening with the nucleus, whereas cytokinesis is going to be division of the organelles and the cell membrane, sort of the cell itself, the cytoplasm and the organelles. So this right here, we have a bunch of little microtubules that are kind of causing the cell to constrict. It's almost like it's tightening its belt. So this cleavage furrow here is going to be, this is the beginning of the cleavage furrow, this is the beginning of cytokinesis. So cytokinesis usually occurs alongside sort of late anaphase, and then it continues throughout telophase and then finishes at the end of telophase, or a little bit after. So this is the cleavage furrow here, and this is late anaphase. So after anaphase, we're going to move into telophase. And so here we can see that once the sister chromatids have been moved to opposite sides of the cell, our nuclear envelope is going to reform. And once the nuclear envelope reforms, then that is going to be sort of the start of telophase. So our nuclear envelope has reformed. You can see these are still attached. They're kind of still being pulled to opposite sides. So this is going to be telophase once the nuclear envelope has reformed. Each of these are going to be two new cells. The cleavage furrow is a little bit deeper now, so cytokinesis is progressing. We have one centrosome in each of these new cells, and then we have a nucleus with a nuclear membrane and a nucleus with a nuclear membrane here. Now finally, as we move here, this is going to be sort of the end of cytokinesis, and we have sort of late telophase over here. And we have got cytokinesis has completed, so you can see that now we have two new cells. They're right next to each other, but they each have a complete cell membrane. Here's going to be our cytoplasm. We have a centrosome in each cell. Then we have a nucleus with a nuclear membrane. Right here, it still looks like these chromosomes are still condensed, like they're still condensed. You can see chromatids here, and because they're still condensed, then I'm going to say this is technically going to be a late telophase, as well as this is the end of cytokinesis. Once these chromosomes sort of loosen up and turn back into chromatin, which will be next, the thing that happens next, then that would indicate that the cell has gone back into interphase to sort of start again, right? Because this is a cycle. Remember the cell cycle.